Welcome to the Bioethics Babe podcast, where we examine the tough questions in human flourishing in light of science, faith, and culture. I'm your host, Darina Grosso-Agnew. Now let's dive in. Thanks so much for listening to this episode of the Bioethics Babe podcast. If you've enjoyed this content, please subscribe to my YouTube channel and your favorite podcast platform, and share with your friends and family. Also check out bioethicsbabe.com for the latest episodes and resources. What if the future of medicine is no longer just about healing the human body, but redesigning what it means to be human? Today, scientists are creating synthetic embryos in laboratories. Researchers are experimenting with three-parent embryos. Human cells are being manipulated, engineered, and combined in ways that sounded like science fiction just a decade ago. So where is the line? At what point does biomedical research move from curing disease to manufacturing, modifying, and quote-unquote optimizing human life itself? Are we prepared for the ethical consequences of a world where human beings increasingly become products of technological design? Today, I'm joined by Dr. David Prentice, President and founding board member of the Science Alliance for Life and Technology, or SALT. Dr. Prentice is an internationally recognized expert on stem cell research, biotechnology, and bioethics, with nearly 50 years of experience as a scientific researcher, professor, academic leader, and policy advisor. He has testified before the U.S. Congress, the United Nations, the European Parliament, the Vatican, and the President's Council on Bioethics on issues including stem cell research, fetal tissue research, cloning, gene editing, embryology, and the future of biotechnology. In 2020, he was appointed by the U.S. Secretary of Health and Human Services to the Federal Human Fetal Tissue Ethics Advisory Board. Today, we discuss stem cell research, human tissue research, synthetic embryos, three-parent embryos, designer babies, modern eugenics, and the deeper question underneath it all. What happens when science gains the power not only to heal human life, but to redefine it? Dr. Prentice, welcome to Bioethics Babe.
Great to be with you. Thank you.
Dr. Prentice, before we dive into these issues that I described in the intro there, can you tell us a little bit about your scientific background and the work you've done in biomedical research over the years?
I grew up being curious. I grew up on a farm. We had lots of wildlife and farm animals and plants and so on. So I was always interested in life and development and how these came about. My research for my doctoral degree, my postdoc, and so on kind of carried through on that. And I ended up focusing on cells, early human development, and all that goes on in terms of that coordination. I just want to say that it's in some way wonderful and miraculous when you consider that we all start as a single cell, a one-celled organism, and we go through all of this fantastic growth and development and cell interactions and signaling, and finally end up with somewhere between 10 and 100 trillion cells as a developed organism. That sort of curiosity and that wonder is what has driven now 50 years worth of research, and even more in terms of just the curiosity about human life.
I mean, that's incredible stat right there. And obviously, that curiosity drove you to spend decades with these questions regarding stem cells, human embryos, biotechnology, bioethics. And could you tell us a little bit more about what made you think also about the ethical questions, the deeply human and ethical questions, in addition to the life sciences, those
Yeah.
curiosity questions about how life begins, how life develops?
Well, when I was a professor back at Indiana State University and a joint appointment at IU Med School at the time, I started getting these questions, some from students and some from friends who had gotten positions of higher positions, even in Congress, about the science, but they also were asking, so what does this really mean in terms of policy, in terms of government, and in terms of bioethics? And I can remember a friend of mine got elected to Congress, and I ended up going up in the summers to help him and answer questions about science. But we really ended up focusing on the ethics. What does it mean to be human? How should you treat other human beings? Even, and again, the focus was on the very earliest stages of human life, things that most people, number one, you can't see that early stage. And people were starting to think about how could we use that early stage of human life. It became very apparent just from the science as well as the ethical positions that every human life is valuable and has human dignity. And we should treat and preserve every human life as valuable.
Yes. And obviously, this is why I'm doing the podcast because human dignity is for everybody, or for all humans, whether they're one cell or an elderly person on their deathbed. So, biomedical research is advancing at a breathtaking pace. Do you think that we are entering an era where science is starting to treat human life as something that we can manufacture, modify, or optimize?
Well, we've already been in an era of manufacture. If you think about simply IVF, in vitro fertilization, and similar types of technologies, where we've taken creation of human life out of the body and put it into the laboratory. That's gone on for years. And we've basically commodified human life in terms of how we look at that life since it's made in the laboratory. We can discuss and unpack that more as we go on. We are entering an era now where we've developed tools that allow us to modify and engineer human life. Your listeners probably are well aware of CRISPR and the CRISPR Cas gene editing enzymes. There are others, but the idea that you could take a human genome, and especially an early human, an embryo, and modify that to your desire, the idea of having a designer baby and be able to, it's usually touted as we're going to treat or prevent disease, but it quickly moves on to we want the type of child that we desire. And so we are looking at that, and that's frankly one reason that we created SALT as an organization of scientists and policymakers is to try and educate people about what's going on in the scientific realm and show the ethical aspects of the science.
Can you give some examples today where biomedical research has already crossed from therapy into enhancement? So, obviously, you started to talk about designer babies. We also had Dr. Tara Sander Leon from SALT to talk about designer babies. Are there other places in biomedical research where this is also happening?
Well, I mean, if we talk about commodifying in our attitude, we can go back to fetal tissue research, embryonic stem cell research, and so on. The modification and the engineering of human life and creating human life in new and scary ways, if you will, is really a factor of maybe the last decade or so. Uh, they tried to do genetic engineering years ago, but it inevitably failed, whether they were trying to do it with embryos or tried to do it with adults. But the new technology with CRISPR and other types of gene editing systems has made this very easy and has made it almost a lure for some scientists to say, well, I can do this, I can do that, I can replace this gene, I can enhance somebody. And newer things that we'll, I'm sure, discuss. You mentioned synthetic embryos. I don't really like the synthetic term. It's a synthetic way to make human beings, but what you've made is an authentic embryo. And this is done Not the old fashioned way with egg and sperm, but by combining stem cells in different ways and different types of stem cells. And it's really fostered an explosion of research, not really meant to help human beings, but just for scientific curiosity. Can I make an embryo this way? Can I grow it in this particular way? So there are a lot of challenges we're facing nowadays.
Do you think the public fully understands what scientists are now capable of doing in laboratories involving human cells, embryos, and reproductive technologies?
And the big answer there is no. And that's another major reason we created SALT. Our purpose is to educate first the public because people need to understand what's going on in terms of these biotechnologies. And it does affect everyone. It's not just the scientists or certain people and certain groups and so on. We're really looking at remaking humanity, for some scientists, remaking it in their own image instead of in God's image. And people need to understand about this and be prepared to weigh in with their own opinions.
Mhm. So, are we having these ethical conversations early enough, or does society tend to react only after technologies become normalized?
Well, I think we are intervening early enough now, but you're right. In the past, if you look at some of the things we've mentioned, aborted fetal tissue research, embryonic stem cell research, the earliest types of cloning, like they used to clone Dolly the sheep, but we're trying to do with humans, we were all reactionary at that point. The scientists started doing these experiments and talking about how they were making human beings and altering human beings. And it was very much reactive trying to stop that. That switched a decade or so ago when they started talking about gene editing and altering the human genome and creating interesting things like three parent embryos. We were able to see what was coming and start to get ahead of that and start to put guardrails in place to prohibit unethical types of research and to foster ethical types. But it's still a challenge because there's so many things coming up that we have to be on our guard and continually reading the scientific literature and watching what's going on so that we can stay ahead.
Are you saying that here in the US, there are now ethical guardrails that weren't there 10 years ago? And if so, what does it look like internationally? What kind of guardrails or lack thereof are there in other countries?
We have been able to come up with guardrails here in the US, and frankly, it was members of SALT. Now, we're a new organization, but a group of people that have worked on these issues for decades. And one example would be there is a law in place. It's a legislative rider at the federal level called the Adderholt Amendment, and it prohibits scientists from doing these kinds of genetic manipulations and gene editing and making three parent embryos in the US. We were able to get ahead in terms of that. There are also discussions about completely doing away with aborted fetal tissue research or human embryonic stem cell research that are going on now. So we're in a better position in the US than many countries are. Globally, there are some countries that are in a good position like the US, but there are a lot of countries, including China, where, as a friend of mine, Wesley Smith, says, bioethics goes to die because they really don't see any kind of ethical guidelines. They pretty much do whatever they want to do. And in a number of those countries, the scientists are really not just pushing the envelope, they have broken through and done some horrific types of experiments on young human beings.
And we've spoken on the show about China before and some of the horrific things happening in China across the board, from the organ trafficking to their the infanticide that was taking place that could, and people being killed, etc. So China has a lot of problems in terms of their lack of respect for human dignity in general,
Yeah.
and it makes sense that even in these biomedical technologies that they will push the envelope and also not respect human life at the earliest stages. So 10 years ago, many of these ideas sounded like science fiction. Which developments do you think would most shock the average person if they realized that this was already happening?
Yeah, well, there are a number of them, but foremost in my mind right now is this embryo models or synthetic embryos or artificial embryos. What these are, frankly, are they're making real embryos, but they're not starting with an egg and sperm, or they're not even doing the old fashioned Dolly style cloning and so on, where they're transferring nuclear material into an egg. They're starting later on in development, and this is done by combining stem cells of different types in different ways, and they actually will aggregate together and form an embryo. It's an embryo that looks like it's maybe 5 or so days past fertilization. So they're not starting early in the first few days, they're starting a little bit later, about the time the embryo might implant into the uterine wall, 5 to 7 days. And they can make these in lots of different ways. And then the other maybe scary part of this is then they will grow them in the lab for days and even weeks. For mouse embryos, there's a scientist in Israel who has grown these types of artificially created embryos halfway through gestation
Mhm.
in little artificial wombs and spinning bottles and so on. And he's flatly said, hey, we ought to do this with humans. We could make, he wouldn't call them actually human beings or embryos. We could make something that we could use to harvest for spare parts. But, the embryos that are made, and they are real embryos. Looks like an embryo, it acts and develops like an embryo. It's an embryo. It's simply that the mechanism for their creation is synthetic or artificial. Really opens up a great danger in terms of cloning human beings and being able then to do genetic manipulation on these newly created human beings. And
Mhm.
the other thing about this is these are really parentless embryos because again, you're not creating them with an egg and sperm. You're creating them from some stem cells that have been growing in the lab for maybe years. So, we're kind of losing an ethical anchor when we talk about we're going to create these embryo models.
Wow. That's so fascinating and terrifying. And we'll talk more about synthetic embryos. Maybe you have a better name for it. But let's first talk about human tissue research, and then we'll talk
Okay.
about stem cell research, et cetera. That way, we're understanding it from the ground up. So, with human tissue research, what does that actually include today?
Well, I mean, if you just say it as human tissue, it could be anything derived from any age of human being. Uh, they've taken tissues, they've taken some of the tissues and made cell lines where they've grown the cells out and continued to grow in culture. And this has been done for over 100 years. So it's not a matter of uh, a scientific problem so much as it is a moral and ethical problem because who you got those tissues or cells from is the real question. And did you get informed consent? Did you get the consent of the individual from whom you're getting the tissues and cells before you harvested them and grew them in the laboratory? Now, consenting issues are really a big problem when it comes to this. Very often, people will get tissues from born individuals, especially adults, and you can sign off, maybe they had to have surgery and have something removed, and it's able to get that. But when we start to talk about fetal tissue now from preborn individuals, there's some real problems that crop up ethically. The main one has to do with fetal tissue from aborted individuals, miscarried or stillborn individuals. Those individuals have died. The parents can ethically give consent for tissues or cells to be taken from them. When it comes to abortion, there's no ethical way to get consent. If you're asking the woman who's going to have the abortion to give consent, that's not ethical. She can't consent to letting that individual's life end and at the same time then being able to take that tissue. It's simply impossible to get true ethical consent. And that's where the real issue has come up in terms of fetal tissue, starting back, frankly, over 100 years ago now. People had talked about, well, it's very young tissue, and so we could use that to do our studies in the laboratory or even to treat individuals and to transplant fetal tissue. It really became more intense in the 90s. There's actually a part of US statute now that talks about allowing transplant from fetal tissue to individuals to try and treat them for various types of conditions. It was again one of these, well, we think it has the potential to happen, and so we're going to allow it to happen. When they finally did the actual studies to try and transplant, and in particular, fetal brain tissue for Parkinson's patients, the results, as the New York Times said on their front page, the results were horrific. Nobody got better. It made many of the patients worse. And if you look back over the over 100-year period for fetal tissue from abortions and trying to use it in not just therapies, but even experiments, 0 people have been helped. It has not cured a single thing. And
Hmm.
in fact, our federal government hasn't funded any of these experiments since about 2005 because it was simply horrific. So it's an area that is rife with problems and concerns and really is going away. Back in January of this year, the National Institutes of Health finally decided after years, we should say, of urging, including from some of our SALT members, we're not going to fund aborted fetal tissue research anymore. Period. End of story. It has stopped, which is
Well,
a very good thing.
yes, that's a major move in the right direction. So, just to clarify, obviously, tissue research, if the tissue is obtained ethically, meaning it was from an adult who had informed consented, then you can do research on it. But when it comes to fetal tissue, it could be ethical if it came from a baby who already passed away from a
Right.
miscarriage, etc. Or, for example, I guess you could get cells from cord blood,
Yeah.
etc. So there are ethical ways to get and to do fetal tissue research. What's unethical is when there's no informed consent and also from an abort because obviously an aborted baby was killed, but also cannot give consent, right? There's no consent there.
There's just no ethical way to get true consent. And you mentioned the ethics advisory board at the federal level that I was on a few years ago. We had people from across the spectrum, scientists who actually did aborted fetal tissue research on there. And we were evaluating grant proposals of people that wanted money to do that type of fetal tissue research. All but one of those grants was denied. And the main issue had to do with informed consent, that you simply could not ethically get that tissue to do that type of research. And I mentioned we had people across the spectrum, including scientists who do fetal tissue research. Even they were voting against those grants because there was the ethics of the problem and the lack of informed consent. And also, frankly, that there are many alternatives, ethical alternatives to do the same type of research and get the same type of cells and products.
Mhm. And before we go to the alternatives, let's dive in a little bit into the ethics around vaccines that were created from aborted fetal cell lines and why we don't have alternatives here in the US for some of those vaccines. And what would you propose be the solution to getting those ethical vaccines? Because some parents are not necessarily against vaccines, but against how they were, where they originated from,
Yeah.
or the things that's in the vaccines. And I know this is a little going a little off topic, but it's part of this. The aborted fetal cell line
It is.
issue and the origins of some of these vaccines is part of this story.
And it's an ethical issue that they're really uh, wrestling with. And so to make sure we're clear on what your listeners are hearing, uh, we talked about tissue before, but I also mentioned cells. And so what people can do, scientists in the lab can take a chunk of tissue, let's say from liver or some other organ, and put it into the laboratory and get the cells from that tissue to grow in the petri dish. It's no longer a tissue. You've dissected it out and separated, and the cells are growing in the petri dish. And you can keep those cells growing for a long period of time. You can also do techniques, and I taught cell culture for almost 50 years as well. So one of the things that we would do with animals and animal tissue is to show that you could get cells from the animal tissue and grow them in the lab. What some people did was take aborted babies and their organs and tissues and grow cells from those tissues in the lab. Some of these have been in culture since the 1960s or 1970s. They will continue to grow for a long time in the lab, and you can do different techniques and so on to keep them growing. You can even alter them to make them essentially immortal so that they will keep growing forever and ever in the laboratory. But you have to think about where these originally came from. The original source was from an abortion, an unethical act where you're actually destroying a human life and not benefiting that human in any way. So there's a unethical connection in a lineage from those original tissues and cells all the way to today. Some of the scientists used those unethically obtained cells or cell lines to actually create vaccines. Viruses in particular, because the way you make a viral vaccine, at least the long term standard way, was viruses have to grow in cells. So you would infect those cells in the laboratory dish with the virus. It would make lots and lots of virus. You would get that virus back out and kill or weaken it. And that's the vaccine that you would get for a number of different types of diseases. But again, you're using this unethical source. Now, the vaccine doesn't contain the fetal cells. It might still have some proteins left over because you broke apart those cells or a bit of fetal DNA. But the real concern is the ethical connection that you used that particular cell from this unethical act to get that vaccine. And unfortunately, there are still some vaccines approved in the US that use those old unethical abortion derived cell lines. There also have been some cases where, and this happened especially with COVID. In fact, our SALT people were the first to really identify this as a concern. They would make the vaccine in some way that didn't involve fetal cells, but then they would test it using the fetal cell lines that we've talked about. So again, you've got this taint, the unethical associations from it.
So,
And that's where the real concern has been, whether it's a few of these vaccines that are still around that use those cells for production or that use them for testing.
so with that, why is it so difficult, or why isn't there no appetite for creating alternatives in the US so that parents wanting to vaccinate their children with vaccines that are ethical can do so. Is there, for example, with MMR,
Hmm.
is there a Japanese version that would be ethical? Hasn't it been replicated here in the US?
Yeah, and you've done your homework. You know that with the MMR, and that's mumps, measles, rubella, they've combined those three vaccines together. And unfortunately, in the US, one or two of those use some of those unethical abortion derived cell lines to make the vaccine. But as you mentioned, in some other countries like Japan, there are ethically derived versions. You can grow viruses in other cells that are not derived from abortion, whether they're human cells or even animal cells, so that you can make this vaccine in an ethical way. I'm not sure that there's no appetite, but it's been reluctance of some of the pharmaceutical companies to come up with something different, whether that's an economic issue with them. Oh, we've got to come up with this new way and a new assembly line and put it together. Whether it's simply a reluctance to answer the call from a significant portion of the public who object to the ethical problems with these vaccines, it's hard to tell. Some pharmaceutical companies have answered the call. A number of years back, some of the polio vaccines used to be made using these abortion derived cell lines. And one company in particular, Sanofi, switched and they no longer use that unethical cell line. They use a different ethically derived cell line. And so all the polio vaccines in the US now are ethically derived, but we've still got some of these we have to worry about. So my point to the companies would be they would actually see acceptance of their vaccines more readily if they were to switch to ethical cell lines for creating these vaccines.
And, if profit is what is driving it, you could even argue that they would make more money by having ethical lines
Yeah,
because more people would be willing to vaccinate their children if there weren't these ethical issues involved. Not necessarily, obviously, there's other issues that they would have to
sure.
consider, but take away that component that holds back a portion of the public from wanting their children vaccinated with that.
Yeah.
So you mentioned that supporters of human fetal tissue research, they will say that, oh, there's been incredible scientific advancements because of it. But you mentioned that there haven't been. Can you speak a little bit more about that? And also, what are the alternatives that have been successful?
And it's hard to come up with any examples that actually show success. One of the things that, and back a decade when they were actually having hearings in Congress on the select panel for human lives and infant lives, they were trying to say that the polio vaccines, the original polio vaccines back in the 1950s, were made using aborted fetal tissue. Simply a lie, because both Salk and Sabin did not use aborted fetal tissue or cell lines from aborted babies. So, they try and point to that as, oh, all these lives were saved and so on. Transplants, as we mentioned, of fetal tissue from aborted babies has never worked. And we're talking about over 100 years. It's more a matter of they would like you to think that there have been these great successes, and this is the only way you're going to treat disease. And that ignores then the huge number of alternatives that are ethical and successful. So, what are some of those examples? Well, the main one would be adult stem cells. We've already hinted, if you will, that embryonic stem cells are an unethical route to take, and that's simply because, as the name implies, you take in from an embryo, an early human being. So, at that point in time, we are the proverbial ball of cells, if you will. That's how every human looks at that stage of our life. But we're still a human being with dignity and value. And you only get embryonic stem cells by destroying that life to get the cells into the dish. It's another example also where there have been no successes with embryonic stem cells. The first ones successfully grown in the laboratory were in 1998. So we're talking over 25 years of human embryonic stem cell research and millions of dollars of taxpayer funds going to that research. And we're still looking at exactly 0 human beings who have been cured with embryonic stem cells. We kept hearing, in fact, early on, oh, in the next 5 or 10 years, we're going to be curing. And there was a whole list of diseases. And then 5 or 10 years later, well, in the next 5 or 10 years, we will cure. I had one U.S. senator tell me that embryonic stem cells had the potential to cure all known maladies. Maybe a little bit of hyperbole there. But again, 0 people have benefited and been cured by embryonic stem cells, where in the meantime, adult stem cells. And when we say adult, I think they coined the term to get them as far away from embryonic as possible. Adults can certainly donate adult stem cells or use them for their own treatment. Bone marrow transplants, we now call stem cell transplants, and it's adult stem cells. But you can take them from any individual after birth. You can take them from lots of different tissues. You mentioned umbilical cord blood and even the solid part of the umbilical cord. You can even take them when they do the amniocentesis on a baby about halfway through development, still inside the womb, and take a few of those cells and be able to grow them in the laboratory and potentially use those for treatments of that same baby. But in terms of numbers, if you had a scoreboard, the score is still 0 for embryonic. It's over 2 million for people that have successfully been treated with adult stem cells for dozens of different conditions.
Wow. So, how has biotechnology changed the demand for human biological material? And I would say
Yeah.
for ethical material. So,
Right,
or and not needing, as you said, these, and we never need unethical science.
right.
And this is part of what bioethics is about, that we can do good science, good medicine, and do it ethically.
And I think the demand has increased almost exponentially, but that's especially because more and more scientists are realizing and doctors are realizing if I want something in the clinic, I need something that's actually going to work and not harm the patient or the donor. And so we mentioned adult stem cells. There's another type of ethically derived stem cell that people need to be aware of. The big term is induced pluripotent stem cells, IPS for short. Induced because you take a normal cell, like a normal skin cell, and you can put it into the lab dish and induce it, kind of reprogram it, if you will, so that it looks and acts like an embryonic stem cell. But there were no embryos involved. You didn't have to destroy anybody or harm anybody to get those cells. There was a Japanese scientist, Yamanaka, in 2006 who figured out how to do this. And it was interesting when he was asked by the New York Times, how'd you come up with this and why did you? He said, I went to a friend's lab. I looked through the microscope at an embryo in there and I thought, we, c- we can't keep destroying embryos for our research and potential therapies. This looks too much like, it's closely related in developmental age to a couple of my daughters. We've got to come up with something else. And so he came up with this IPS cell technique, won the Nobel Prize for it as well, and he should have. But you can derive these cells and use them. They're starting to look at potential treatments. It's a fairly new, but you can also use them in the laboratory for a great number of experiments and challenges and looking at normal development versus abnormal development because you can make these cells from anybody and almost any tissue. So you can study a disease in the laboratory.
And since we're already discussing stem cells to this extent, could you talk about, in terms of these adult stem cells. So you said obviously it could be from a child, but these are more mature cells and from a
Mhm.
born child, and so they're considered adult stem cells. But how can these be used, and how have they been used in real world therapies in ways that embryonic stem cells can't even hold a candle to it?
Well, as I mentioned, there are already over 2 million people around the globe who have benefited versus 0 for embryonic. Besides the ethical problem with embryonic stem cells, there is a practical issue. These cells like to grow, and so when they inject them into the lab animals, they usually form a tumor. They don't help
Mhm.
the animal, and you wouldn't want to treat a human being with those cells. The adult stem cells are a little more manageable. They're more controlled by the body and by their own controls. And so, whether they're making new tissue directly or stimulating repair of a tissue, they've been used for dozens of different conditions. We actually, Salt is managing another website, Stem Cell Research Facts, where we have profiled a number of patients treated with adult stem cells. And this is for things like leukemias, sickle cell anemia. One young lady, uh, she was treated when she was about 20 years old, had lived her entire life in pain because of her severe sickle cell anemia. She was treated with adult stem cells from umbilical cord blood. So the baby's born, we cut the cord, don't throw that away. Keep that umbilical cord blood stem cells on the solid cord and use it to help somebody. She was treated with cord blood stem cells. Her sickle cell anemia is gone. And she was inspired to become a nurse because of the health benefits she had received. She wanted to help other people and so on. Even genetic conditions, like there's a condition called Krabbe's disease. It's a neurological problem, genetically derived, and little kids who have this usually die within about the first 2 years after birth. We profiled a little cutie named Reagan Moore who was born and diagnosed at birth with this and then treated with, in her case, because it's genetic, like sickle cell, with umbilical cord blood stem cells that would match. uh, she started school a couple of years ago. I mean, she still has some difficulties. We're not saying it's 100% cure for her, but we're talking about it's added years to her life and the joy that she gives everyone around her who's simply exposed to her. And we can go on and on. Like I said, over 2 million people and dozens of different diseases treated for, whether it's heart damage or stroke or a number of different conditions. These are the cells that work, and they work because they're more manageable, and they are, I think, because they're ethically derived. We're seeing a correlation here that if you derive something ethically, it's going to have a beneficial effect, whereas the unethically derived research or experiments really does not benefit humanity.
Is there still a push, though, for embryonic stem cell research? And if so, why? When all the benefits are with the adult stem cells and when all these alternative
Yeah.
approaches exist, what's pushing that agenda?
The lure of money. If I'm a scientist and I'm convinced that I can make this work, and there are a number of scientists who think they're going to be the one to finally make it work if they get enough money to run their lab and do all of these experiments with embryonic or even with the fetal materials. But it's still not panning out. And if we want to be good stewards of taxpayer money, we will cut off that spigot, as we already have done. The NIH has done for aborted fetal tissue, and they frankly are taking comments. They took a bunch of comments that they're looking to do the same thing with human embryonic stem cells to stop wasting money and wasting lives. Let's not forget that part of it for unethical research that is not benefiting a single human being and put that money towards adult stem cells and these other ethically derived types of therapies that are already bearing fruit for human beings.
What do you say to researchers who argue that embryos used in research would otherwise be discarded through IVF?
So, what they want to do is go ahead and kill people because that's what you're doing. If you destroy those embryos, you're ending a human life simply because you don't think they have any value or any future value. And you could use that same argument for newborns, for teenagers, for any human life that I don't think it has value unless I'm going to use it and exploit it for my own desires and ends. It's simply not a valid reason to continue a research line that relies on the destruction of a human life.
Well said. And obviously it's a way of justifying what the IVF industry is doing anyway. It doesn't make it right. And using embryos from that are stored from IVF cryopreservation is also not ethical. So finding a quote unquote use for it is not justifying it, and it still doesn't make it right. So we discussed earlier a little bit about synthetic embryos, but this concept is so new and cutting edge. Can you explain how we actually what synthetic embryos are and
Mhm.
how can we actually get an embryo without using sperm or egg?
Yeah, and it's an advance in quotes, if you will, for biotechnology, another way to make a human life, a human being, but without starting with egg and sperm. And I do want to point out we should maybe change our terminology. These aren't synthetic embryos, they're embryos made synthetically. So there's an artificial or a different process. Some of the scientists have called them embryo models. That might be the best term to use for now, but even there, they're trying to say that they're not really embryos. They're just trying to model normal embryo growth with something that they've made in the lab. And how do they make them? It turns out that early in our life, early in embryonic development, our cells know how to get together to form that early embryonic stage and then continue to develop from there. And so, what they've figured out is they can take stem cells, and it could be embryonic stem cells, it could be adult stem cells, it could be some of these iPS cells, but they can treat them in certain ways that they will come together, aggregate into that proverbial ball of cells, that, if you will, but they will form a normal looking embryo. And they've done things like try to withhold certain cells that might form certain parts of the embryo, but they're still pushing. And in fact, they will gloriously claim in some of their scientific papers that we've made something that looks exactly like an embryo at this stage of development. Oh, by the way, it acts just like an embryo at that stage of development. We ought to call that an embryo because that's what it is. It's made in an artificial process, and it actually starts its life farther down the road than we would normally start. So you're not starting at a single cell like you would with fertilization or even the old fashioned ways of cloning, like were used with Dolly the sheep. You're starting the embryo at about 5 to 7 days after what would be the normal conception. But again, these cells come together, it looks like and it develops like a normal embryo. And they will continue to grow these embryos in the laboratory, pushing the envelope again there. The short story being they have grown some of these to the point where they have beating hearts, and you're starting to see other types of organ development. That's an embryo. It's a human life. Even if you have designed it to have a defect, what you've done is design a defective human being, one that you have purposely introduced some sort of problem, maybe a genetic issue with, but it's still a living human organism. And we need to start looking at this in terms of this is just another way to make an embryo. It's, in fact, in a scary way, a better way to clone. And as I mentioned before, these are parentless embryos.
Mm.
That might be the best phrase to use to describe these because you're not starting with a sperm and egg of any kind. You're starting with stem cells that may have been grown in the lab for years, and you're putting them together to start a new life, multiple new lives.
Wow. So with and I think I'll just call them lab made embryos. Maybe that's a better term than synthetic. Um, but so where does that DNA information come from? It comes from the cells that were used that
Yeah.
belong to somebody. So obviously, somewhere down the line, they're genetically related to somebody.
Yes.
But how does basically you can even make them from skin cells, right?
Yeah, you can make them from pretty much any cell. If you were to take that skin cell and make the iPS cells that we described before that Dr. Yamanaka came up with, again, those look and act like embryonic stem cells, but they're not made from embryos. They're made from a skin cell or a liver cell. In fact, the early days when they were first figuring out how to make these in the lab, there was one paper where the scientist went over and pulled a hair from his lab partner and used the hair follicle cells to make these iPS cells.
Mm.
You can make them from virtually any body and almost any tissue that you can. You grow them in the lab and then you treat them in such a way and put them together in the lab dish, and they will spontaneously come together to form a lab made embryo.
Are there limits to so basically they're creating these embryos and then they're destroying the embryos. So you're killing these embryos in the lab. Are there limits to how long they're allowed to live? Obviously the creation of them is problematic and the killing of them is problematic. And if there are no limits and if what happens will they will we reach a point where they're going to be gestated? And if so, do we know what kinds of potential health conditions, etc., lab-made embryos might have? What kind of information do you have in this new world?
And great questions, and for which we don't have a lot of answers yet. In terms of limits or regulations or laws, there's very little. In the US, basically none. In some countries around the globe, there has been legal limits called the 14-day limit was instituted back in the 90s when people were talking about using human embryos, created the old-fashioned way with egg and sperm, but using them in the lab and growing them and doing experiments. And the compromise, it wasn't really a compromise, it was a way to get at more human research. But what scientists proposed and was agreed on was that, well, you could grow human embryos in the lab, but not longer than 14 days. And at that point, you have to destroy them if you could get them. And it was touted as this great ethical compromise to prevent a lot of human experimentation, especially on embryos. But it allowed them to grow them in the lab for up to 14 days. Now, it was an easy limit to abide by because for years and years, nobody could grow a human embryo in the lab that long. If they could get them to 10 days, they were lucky. And in most cases, even 7 or 8 days was the best they could do. So they could make themselves look like they were being very ethical and they weren't doing too many experiments. They were still growing human embryos for a few days and destroying them.
And now, oh,
Yeah, well, about 10 years ago, a couple of different labs around the globe found out that they could grow human embryos longer and longer. And they grew them right up to the 14-day limit. It's a law in countries like the UK. It's a suggestion in the US. There are really no legal limits in the US. And, maybe just out of conscience, they stopped their experiments at 14 days, but immediately the proposals were, well, let's just do away with the 14-day limit. What if it were a 28-day limit or a 35-day limit? And there's a group, it's a self-concocted group from the International Society for Stem Cell Research. They are promoting anything that has to do with stem cells and especially embryo destructive research. And they come out every once in a while with recommendations. Well, after this discovery that you could grow embryos longer than 14 days, their recommendations were not 28 or 35. Grow them as long as you want, as long as you can justify it scientifically. Wink, wink, which means do whatever you want. Now, the countries that actually have legal limits are working to change that. But in the US, again, there is no legal limit. And so now we have them growing these embryos in the laboratory for weeks at least until you have a beating heart and other tissues starting to form, and proposals to come up with some sort of artificial womb that you can grow them even longer. A scientist in Israel has come up with a system of some spinning bottles and been able to grow mouse embryos halfway through mouse gestation, and proposed that we do the same thing with authentic human embryos as well as these lab created embryos. Grow them, do genetic experiments on them, maybe even grow them to the point where you could start to harvest organs and tissues for transplant. So, this is really the scariest part of modern technology, along with the gene editing, remaking humanity and creating an entire class of human beings whose purpose is simply to be destroyed and be a commodity for spare parts.
It's really scary that there are no limits, that this is happening already, that these embryos are created, destroyed at will. And so, besides an artificial womb, could they conceivably be implanted the way that IVF embryos are implanted?
Definitely.
In
Because you actually start them just a few days before an IVF embryo normally would be transferred to the womb. And they form again the structures just like you would see in any IVF embryo. So they have the ability as an embryo, given the right circumstances, to implant and begin gestation, whether that's in a human womb, whether it's in an animal womb, or whether it's an artificial womb. And again, there are basically no guardrails to stop these types of horrific experiments on young human beings.
has that been tried yet? Implanting them, transferring them to a womb, whether it's a artificial womb or an animal womb or a human womb?
To the best of our knowledge, no, although the Israeli scientist that we mentioned has tried to do some of those experiments or combine human and animal cells together as an embryo, human animal chimeras, and grow them. It hasn't worked very well. The current limit seems to be simply being able to have an oxygen supply, a blood supply. So when we implant into mother's womb, we immediately form a bond. We form that placenta that allows us to get oxygen and nutrition. They haven't
Mhm.
come up with an artificial placenta yet, which is really the key critical step for a true artificial womb and what we would call ectogenesis, growing a human being completely in an artificial environment instead of a normal womb. But they're still working on it. In fact, there were a couple of papers just recently where instead of just using the typical petri dish, They first put placental tissue into the dish and then added this lab created embryo, and it acted like it was starting to implant. So, more to come, and more for your listeners to be aware of, that they need to be on guard about the great scientific advancements that they're going to hear of that really are degrading human dignity and destroying human lives.
And what can be done, though, to put a stop to this? What kinds of laws need to be passed so that it sounds like it's a free for all? So, right now, someone could implant a lab made embryo into a woman, transfer, and then potentially grow this human being. And we don't know exactly what the results of that would be because we
Yeah.
haven't seen it and the effect that might have on humanity. And in terms of how that changes our our uh our makeup, right?
It really is a problem, and, we need some sort of guardrails, whether those are legal. Obviously, the best guardrail is a moral guardrail where people's conscience says, I might be able to do this kind of experiment, but we should not do it. That age old question, not can I do it, but should I do it? Lack of moral guidelines and formation for a lot of scientists mean that we do then need some sort of legal limits that just say, no, you can't do this kind of thing. We've currently got some partial limits. They're more a matter of you can't use federal taxpayer funds to do these experiments, but that leaves private and state funds. And a number of states are interested in what they can get out of commodifying human lives. So we really need some, not just federal, but even international laws, even a moratorium that would just say, let's stop for now and really think long and hard about whether this will actually benefit humanity or whether this degrades everybody because you're degrading certain members of the species.
And this goes back to why bioethics is important and even the
Yeah.
principles of bioethics because the ends don't justify the means. And this was in our first episode and a message that we've been saying throughout. Even if we could do this, as you said, ethically, it's not the right thing to do. Even if it advances science and
Right.
technology, we have to find ethical solutions to our medical problems. And there should be a moratorium, and not just for a time. There should be a moratorium on unethical research like this.
Yeah. Some sort of hold, some sort of stop, just for people to start to think about things like this. And there've been proposals even for international moratoriums, but we need to move ahead and make those reality.
Yes. And you already mentioned that these are real embryos, even though they're lab made and that they look like an embryo, they act like an embryo. But I think that there's still a disconnect. Some people might still see them as, well, they're just made in a lab, so they're not really real. Can you talk about the characteristics of these embryos and how that differentiates from embryos made the way that embryos are usually made and to kind of dispel that idea?
And I think, we used to say, well, an embryo, how do you define an embryo? It's something created at an egg sperm fusion and it has a human genome. Well, you're making these lab made embryos and they're not starting at egg sperm fusion and they're not starting at a single cell. They're starting further on. But I think you can go back and use those same principles just at a later stage of life. So, human genome is obviously important. That's the key consideration, if you will, but not just having a human genome, it's being an organism. So, my skin cells each individually have a human genome, but they're not an organism. You couldn't put those into a lab dish or into a womb and get another human being to grow from them. So there's a key part of the definition that involves being an organism. Now, these organisms, you're starting farther down the line than we would normally see, but there's a series of embryological definitions for what a human embryo looks like and acts like at each step along our first few weeks of life. They're called the Carnegie stages. And I think you could easily say this looks like its morphology is such, and it acts like its developmental progression and the things going on are the equivalent of a normally or fertilized human embryo at that Carnegie stage of development. But again, it's a matter of this is an organism that is living, growing, developing, and we need to recognize that that's a human being too, and he or she deserves our respect.
Hmm. And at what point would you say that lab made embryos be recognized as possessing moral significance?
I'd say right now. So, you know, instead of the old 14-day rule, my colleagues and I suggest a 0-day rule that from the very beginning, no matter whether they're starting at a single cell organism or they're starting at a 100-cell organism, the key point being this is a human organism. This is a human being, a human life. And as soon as you are conceived, whether again that's at a single cell or hundreds of cells, you are now a human being, a human organism, and you deserve respect and protection because you're a valuable human life.
Yes, absolutely. And you already mentioned that scientists are doing this one to test the limits, but also because potentially they could use parts of these embryos eventually for organ transplants, etc., which doesn't make it right. Again, the ends don't justify the means.
Right.
We're not just a series of parts that can be destroyed and used at will. But what are these researchers hoping for? Are those the reasons? Are there other reasons that they're doing this?
I don't know exactly their thought patterns and their desires, but I think we can presume a number of them. And one is, as scientists, they're curious. They're just wondering whether I can do this. I want to be the first to do it, of course, is another motivation for a lot of scientists. Being able to, frankly, play God and be able to create life under their own conditions and using their own guidelines and models. And frankly, economics weighs in again for a lot of these. The motivation might originally be phrased as we want to be able to supply organs for transplant for all of these people that can't get them, or we want to be able to discover the cause of a certain disease or a genetic condition and be able to alleviate that. Some of them have also talked about we want to be able to study how embryos grow and implant so that we can help pregnant women or make IVF better and so on. But in the end, how is that going to be put into practice? And are they going to benefit financially from it? I think, you know, that's a big motivation for a lot of these people.
Yes, and when we had a Dr. Torsten Trey on the show to talk about organ trafficking in China,
Mhm.
where in China they're killing people specifically for organs. This is no different. Where
Right.
they're just killing embryonic people for organs, and that's what they're going to try to do with this. So that's not resolving the ethical issue
Right.
of getting organs. Obviously, organ donation, there are ways that that happens ethically. There are ways that that's rushed, and
Yeah.
maybe people are still being killed for their organs. But this is not a solution from an ethical perspective to that issue.
Now, this is specifically creating human beings as commodities to be used experimentally and for their parts.
Well, let's turn now to three parent embryos and germline engineering.
Okay.
So, again, for the sake of the listeners here, what exactly is a three parent embryo? How is it created?
Okay, and let's back up one step and say, what's the rationale to even go this direction, other than the scientific curiosity about, I wonder if I could do it. And so this was the proposed solution to a problem that's called mitochondrial diseases. Within every one of our cells, including in the egg, there are these little energy factories called mitochondria. And while most of our genetic material is in the nucleus, and certainly the genes that determine, eye color and hair color and all of these various traits, these little energy factories, these mitochondria also have a little bit of their own DNA. DNA can be mutated. They're energy factories, and so the cells relying on them, and if they have mutations that then cause problems with energy production, these can lead to problems for not just that cell, but the entire organism, the human being, that has those types of genetic mutations. It's a genetic disease, if you will, just as having that genetic mutation in our nuclear DNA can be a problem. And so, because they're in these little energy factories outside of the nucleus, just in the cell cytoplasm, their proposal was, let's transplant some good mitochondria that don't have mutation into that cell. Well, they tried that and it didn't work too well. So then the next idea was, well, let's transfer that nucleus out of the environment with the mutated mitochondria into a new egg that has normal unmutated mitochondria. And so you take an egg that has the mitochondrial problems, you transfer the nucleus into a new egg where the mitochondria don't have the mutation. Now you've got DNA from two different mothers because you get all of your mitochondria from your mom. So all of the mitochondrial DNA. So you've got an egg that has the transplanted nucleus from mom number one, the mitochondrial DNA from mom number two, and then you add dad's sperm, and now you've got three parents. Well, theoretically, this maybe sounds like it's a workable hypothesis. I do want to point out that you're transferring the nucleus, which is the definition of cloning. Somatic cell nuclear transfer is what they did to create Dolly the cloned sheep and all the cloned cats and rats and so on we've heard about. So you're doing the same type of procedure and then adding the sperm. Or another way to do it is you create two embryos and you kill them both and you transfer the nucleus from one into another that has the normal mitochondria, but you're still dealing with cloning in terms of nuclear transfer and destroying young human beings. It's a large scale type of genetic engineering. They've tried this. It didn't work out the way they planned. And your listeners may have heard within the last year or two that in the UK, they actually approved this in Parliament for what they call the HFEA, the Human Fertilization and Embryo Authority, which is the big sort of science embryo regulator in the UK. They never met an embryo experiment they didn't like. And so it was quickly approved for them. They went ahead and gave licenses to do this type of procedure. It's a cloning procedure. There have been a few babies born. The results were equivocal. It turned out that they didn't get rid of all of the mitochondrial mutations. This is one example where we got ahead of the unethical science in the US, where we put that legislative rider called the Adderhold Amendment in that says you can't do this in the US. So there was a fertility doctor in New York State who went to Mexico to do the procedure, did implant the little embryo that had gone through the genetic manipulations. The child was born. Turned out he still had the mitochondrial mutations.
Mm.
And it's a condition that they can't get away from, even with all of the unethical manipulations. It just doesn't work very well. There's a lot of embryo death again, like in a lot of these procedures where you're manipulating young human lives in the lab. And there are alternatives that you can use instead of trying to genetically engineer these young human lives.
Mm. So if they're not even successful in this, is there still a push for creating three-parent embryos, or because of the results, have they slowed down uh, in this route?
That's a good way to put it. You might say that they have slowed down, but there's still some labs, not in the US, but in a couple of other countries where they are still trying to do this. Now, what they're saying is it's not to get rid of the mitochondrial disease. They want to do it for other women who might be older and try and help their IVF success. Hasn't worked very well there either, but there's still people that want to experiment with embryos and do genetic engineering on young human lives.
Hmm. Does creating a child with genetic material from three individuals fundamentally change human reproduction?
It it doesn't change human reproduction, but it changes our attitude toward creation of children, because here it's proposed as a way to get around disease. And oh, if we just do this technological treatment, we can engineer your child so that in this case they don't have the disease, but the extension will continue. So maybe they have this trait, or maybe they don't have this trait, and so on.
So what are the ethical alternatives to this? You alluded to that.
Well, it turns out that you can actually transfer, even into a born individual, new mitochondria by adding our old friend, the adult stem cell, that you can transfer some of those good unmutated mitochondria and alleviate a lot of the disease conditions. It's a work still in progress. It's not, you're not going to run down to the clinic and be able to get this right away. But they've shown in a number of experiments that this is a workable way around. The other way that looks promising, again, not clinic ready yet, but working in experiments, is to use gene editing, to use CRISPR or a different type of gene enzyme and get rid of the mutation in the existing mitochondria that have the problems. So there are other ways to get at this, and they don't involve creating human beings and manipulating them. They involve actually trying to treat individuals that have certain types of diseases.
And that's phenomenal because then you're also not introducing a third parent
Right.
DNA information into you. All of those things are not even part of it. So there are ethical solutions and avenues for helping people with this kind of mitochondrial disease, et cetera, as you
Right.
mentioned. So now that we're talking about gene editing, can you talk about ethical gene editing and unethical gene editing? Because you have both. Again,
Yeah,
I look at medicine and science as uh, it's we can do it ethically and we can do it unethically. So and there's always going to be an ethical solution. So and if we haven't found it yet, that's what the scientists should be looking for.
that's
But
right.
even with the gene editing, we can use it well and we can use it in a way that's unethical. So please help us understand what that looks like.
And people should think of these as tools. And tools can be used in an ethical way or in an unethical way. And a lot of times that depends on what your target individual is that you're going to try and use this tool on. So, gene editing, people should not think this is all unethical. There are ethical ways to do editing that really benefit human beings and humanity in general. And then they're unethical. And those targets for the gene editing are the real key here between the ethical and the unethical. Let's let's deal with the unethical first, and that comes back to embryos and the idea that you're going to do this editing on an egg or a sperm or an early embryo and change their fundamental DNA sequence. It's termed germline because that's the DNA that's passed on. I like the term heritable better because once you make that change so early on in our lives, every cell of that individual will have the changed DNA, which means you can pass it on. Your progeny can inherit that DNA change. And there's a real problem here with scientific hubris in that, oh, we think, well, we're just going to change this one little gene and we'll do away with a disease. This person won't have it in the future and so on. The tool is not that good. It is not 100%. And you can get what are called off-site clips and cuts and changes, where instead of the one gene that you're trying to deal with is not the only site in the DNA that's changed, but there are other sites. And the other part of this hubris is thinking we totally understand human genetics at this point. It's pretty complex, and we really have no clue what we're doing. We think there might be 20 to 25,000 genes in the human genome. But reading a paper the other day, they were saying, well, there are probably other genes kind of hidden in there that we don't really detect under normal circumstances. And then at another level of complexity, because it's not just one gene does this and one gene does that, they interact with each other and their products interact. And that's what helps our bodies keep running and our cells keep running. So, perfect example. There was a Chinese scientist a number of years ago who announced to the world that he had done heritable gene editing on two little girls as embryos. He did the gene editing in the lab, but he actually then transferred those little girls' embryos to the womb. They were born. And so he's announcing this to the scientific community and the world, and they were all uniformly negative in terms of that. Some of them, I think it's because they wanted to be the first one to do it, but uh, not really knowing the outcomes. And as they started to study, his initial target was going to be a gene that allows your cells to be infected with the HIV virus. He said, well, if I just knock that out, these little girls will never be able to be infected with HIV. Well, it turned out, again, the tool was not 100%, and it didn't work perfectly in both of the girls. So he didn't even achieve his initial goal. But the other problem is our hubris in thinking we know genetics. Even if he had attained his goal and they could not be infected with HIV, come to find out that the gene he was working on also has other roles and interactions. So he probably made them more susceptible to influenza, West Nile virus, probably changed their cognition and shortened their lifespan.
Wow.
We really don't know what we're doing with heritable gene editing, and that's definitely where there ought to be a complete prohibition, if not a moratorium. On the
And I mean,
ethical
with
front,
these
yeah,
girls, right? They now have this. They will pass that on. Those
right,
gene edits they're going to pass on when they have children.
yeah. So, he's created a problem that we really don't know the extent of the eventual problem. And whether it's those two little girls, whether it's anybody else where we think we are smart enough to know how to control this and do this, you're passing it on. It's not just affecting that individual, you're passing it on to future generations with completely unknown outcomes. So let's Go
And
ahead.
you can't put that genie back in the bottle, right? These
Now,
these
once
girls, they're going to have children, and their children are going to have children that will continue to have those genetic modifications, and anyone who has children in that line,
yeah,
that's going to be passed on. And we have no
it
idea
will.
what influence this is going to have on all generations, and eventually, if this continues and more of this is done, it could completely change the human genome.
It really is an existential threat to humanity in general because it doesn't just affect that individual, it affects future generations. It could potentially even affect what we think of as human and the human genome. So,
Mhm.
heritable, big problem, big concerns, something that we really should steer clear of. And there have been scientific organizations and ethicists and scientists around the globe that have all gone, yeah, we need to just shut that off. It's not going to benefit people. And there are other ways to get at disease treatment and prevention, and that's what's called somatic gene editing. Basically, not dealing with something that would cause heritability, but dealing with just treating that individual. And if you do this later in an individual's life, you can even do it late in gestation, but certainly once you're born, you target just that individual. And what you're doing is now actually treating that individual's genetic disease and problem. And it's already been shown to work. So there were about a year and a half ago, two new genetic therapies for sickle cell anemia that, and they use the C word. You can cure them of sickle cell anemia by treating them with this genetic therapy. There are other types of blood diseases and other conditions they've been working on, and other inherited genetic diseases. Your listeners might have heard news about a year ago of baby KJ. He was diagnosed still in the womb with a particular genetic condition, and they made a genetic therapy just for him so that when he was born, they started the treatment. He's now a little over a year old and doesn't have that condition anymore. So there are ways to do this that don't endanger humanity in general and can actually help individuals. But that's with this more targeted gene editing and genetic therapies.
Wow. And in terms of, just for clarification, so the germline or the heritable gene editing, that's the unethical one, that's the
Right.
problematic one because it can be passed down. We have no idea what we're doing and how it's affecting the human genome. But with somatic gene editing, which is specifically for that person and it's not passed on to their children, are all are all somatic gene editing techniques ethical, or even within somatic gene editing, are there some that are problematic?
I think what you want to do is carve out a somatic gene editing technique that targets eggs or sperm. So if we're trying to treat somebody, you're going after some tissue, some organ where there is a genetic problem. But if they were to take a born individual and say, we're going to make a genetic change in your eggs or sperm, now you've made it heritable, which comes back to the problem that we don't know what we're doing there anyway. And by the way, what disease were you trying to treat by going that route?
Mhm. So, is there an meaningful ethical distinction between preventing disease and enhancing or designing future children?
Yeah, I think when we talk about enhancement, you're talking about trying to add other characteristics or increase other increase certain characteristics that are normal to humans. And when we treat, we're trying to bring back to the norm. When you enhance, you're trying to go beyond the norm, whether that's I want my kid to run faster or to be able to see with night vision or some of these kind of science fictiony things, which, again, science fiction becomes science fact so soon if we're not cautious about applications. But I think you can make that distinction.
Mhm. And of course, I had Dr. Tara Sandra Lee with SALT to discuss designer babies. What's the most concerning thing for you about the new trend of pursuing designer babies, quote unquote?
Yeah, I think for the public in general, the sort of siren song that we're going to help you have better children. In fact, that's some of the advertising. There are several companies that now advertise, we will help you pick your best IVF embryo so you can have a better baby. There are even one or two companies now that say, and we will help you gene edit your embryo so that you can have better babies and so on. It's very elitist. It's not actually helping human beings or humanities. It's trying again to create this distinction between the improved human being, if you will, and others. And it reminds me of a phrase from James Watson of Watson and Crick, discoverer of DNA, a couple of decades ago, who was promoting this, and his phrase was, if we could make better human beings by knowing how to alter genes, why shouldn't we do it? Well, I think there's a pretty obvious answer because we don't know what we're doing. And who's to say that that individual is better or is not? Uh, if you look at individuals that have certain genetic conditions, Down syndrome in particular, that additional chromosome actually contains a lot of extra love and joy than we normally see from individuals. But we kind of had the attitude that, well, that's not normal and we should try and get rid of that. No, we need to let human beings be human beings in all of their great variety, and we need to value every human life.
Yeah. Amen to that. So, eugenics was often justified in the name of science, progress, and quote unquote reducing suffering. Are there parallels today between eugenics as we've known it and when people are now trying to create their quote unquote designer babies?
Definitely that. I mean, in the old days of eugenics, if you will, it was a matter of breeding, almost like breeding cattle. I grew up on a farm, so you would try and limit interactions, so you only got certain types of crosses, and that's what was done in these older days. The better family farms and the better family types of discussions and so on, where they would actually have families bring their kids and say, "Oh, my kid's better, and look how big a smile he is," or "She's so pretty," and so on. Well, we've just taken that to a genetic and a molecular level nowadays, and we think we've refined that so well. But again, there's just an attitudinal problem that, well, this person would be better. My child would be better if we changed this gene or if we did this or that. And again, it's our attitude that is the biggest problem, that we think some people are better or some traits are better instead of reveling in the great variety of humanity.
Yes, and as you mentioned, these were quote unquote fitter family contests at fairs and
Yeah,
etc., where they compared children and adults, etc. So, and we're really just doing that in a different way, as you said,
right.
more at the genetic level now. So, how does prenatal screening intersect with concerns about modern forms of eugenics?
Well, it's just another step along the way. So you go from just breedings and interactions, if you will, to, well, but now we're trying to catch, we're doing quality control, if you will, if you're doing prenatal screening. It's a eugenic type of technique because despite what might be said about, well, we just want to know what to expect and so on. What are the usual outcomes if someone is diagnosed prenatally with a particular genetic condition? Go back to Down syndrome or the other trisomies. Iceland announced, oh, they've eliminated Down syndrome. No, they eliminated Down syndrome people. So diagnosis prenatally usually is a death knell if you have one of those trisomies, despite the fact this is just a human being with a slightly different genetic variation. And those individuals, instead of the negative attitude we have nowadays, especially in the medical community, they would usually advise that that individual just be aborted. And the screenings have gone from whole chromosomes to down to individual genes and mixes of genes, what's called polygenic screening. But again, you can't predict how that's all going to turn out and try and pick the best embryo and do that. And of course, the next step is, well, now we're going to tweak that genome and we'll do a little gene editing and we will deliver you the desired child.
Yeah, it's really quite crazy what has happened and what can happen. So could biotechnology unintentionally create new forms of inequality between genetically selected and the quote unquote unselected, the ones who are naturally made?
Yeah. And I don't know that it's unintentional
Hmm.
because certainly these companies that are promoting the embryo prenatal screening, genetic screening, and definitely the ones who want to gene edit your embryo, those are intentional. They're out to make money off of selling you on this idea that this embryo, this child is better than another, or will correct the defects. There's an old science fiction film called Gattaca, Gattaca, came out in the 90s, where it was a future society, though not that far away from where we are now, where if you wanted to give your kid the best advantage, you know, not just hire tutors or get them a trainer or whatever. You had them gene edited as embryos. And it goes through uh, the main character being created the old fashioned way. What one of the other characters said, oh, you're a godchild, versus his brother and others in society who had that genetic advantage and got all of the societal advantages and showed the humanity though of the unedited person. And that's where these things are headed if we talk about gene editing or genetic screening prenatally and eliminating those that we don't think are the better individuals and promoting this elitist idea of gene edited being the proper people to have.
Right. And just to clarify, there's a world of a difference between trying to prevent disease and eliminating a person with a disease. And
Right.
so, obviously, as you mentioned, there are some forms of gene editing, the somatic gene editing, where you're trying to cure a disease and it can be done ethically. But when you're altering the person or killing the person because of a disease, it's then it's a problem because you're making a quality based judgment of that person's worth
That's right.
when we're killing people because of a disease, or for example in the case of IVF where different embryos are made and then the selections are made based on certain qualities. There's a judgment on the part of the lab technicians or even the parents as to what they would want or like, and as you said, it may not pan out. They might say, well, this is not the baby I quote unquote ordered, and there's no refund process on this, right? Because we're talking about human beings, and even when you do the screenings, it could be that the screening just shows a capacity for disease, and it may not
Right.
happen that way, or it may not, and then it shows up later. It's where when we start to try to design human beings, design children, there are a lot of issues that can come up. So At what point does medicine stop treating disease and start redefining what kinds of humans are acceptable?
I think we're looking at some examples now in terms of whether it's genetic screening prenatally or even postnatal screening and the sort of attitude that we might have towards individuals, not just in terms of their exterior appearance, but their internal genome. And we say, well, you've got this particular gene and so on. Well, that's not as good as my gene or whatever. It really does come back to that attitude, and especially among the medical community, but larger as well in terms of society. We mentioned Down syndrome and prenatal screening there. And what we found is as they've become more efficient, should we say, at doing the prenatal screening, whether it's Down or one of the other trisomies, we find that the medical community in general, their attitude, and they pass it on to the parents and to society, is that those individuals don't have as much value. And that becomes then a societal burden, actually, in terms of how we view other individuals. I actually came across a paper not too long ago where they'd come across a burial ground. I think it was in Spain. And they found several individuals, infants or young individuals who had been buried. And they noticed that when they did genetic analysis, some of these individuals had trisomies,
Hmm.
like Down syndrome, trisomy 18. They were buried with great fanfare and love. They were adorned and obviously loved by their group, not just the family, but the entire society at that point.
Hmm.
And we've obviously gotten away from that where we're going to consider somebody with a trisomy or some genetic condition as less valuable. These people were honored, and that's where we need to be. Every human life is valuable.
Absolutely. And we actually did 2 episodes on prenatal diagnosis, one with Dr. Robin Perucci to discuss what supports there should be for parents, what supports there are for parents who are facing a prenatal diagnosis. And then I had Senator Rick Santorum on to talk about his daughter Bella, who has trisomy 18. They were told that she wouldn't live for very long, and here she is, just having celebrated her 18th birthday.
Yeah.
They never thought that she would the doctors never thought that she would make it
Right.
as far as she has, but she had the support and the loving family and community to help her thrive with a trisomy 18 diagnosis. And so obviously those are such great examples of how we should be treating all of our neighbors in the
Yes.
human family, regardless of whatever condition they have, right? We have uh, handicap parking spots right up close in to the store, to whatever building, and yet before they're born the doctors are telling parents to abort those children.
Right.
And so it's this dichotomy of how we treat people with disabilities. So let's zoom out a little bit and talk about what it means to be human in light of all of this. So beneath all these debates seems to be the deeper question: what does it mean to be human?
And we touched on this before. So I think it's a matter of a human genome, but a human organism. So an individual, a life, and that could be a single celled organism, that could be 100 trillion cells, or somewhere in between that number. But the idea that this is a living organism, a living human being, and that simply by itself is enough to recognize you as a valuable individual, no matter what your various traits are. Simply being that human organism is the key to respect.
Mhm. Is modern biotechnology still fundamentally ordered toward healing the human person, or are we beginning to reshape humanity itself?
Great question. I think to a large extent, it is still focused on healing. In a sense, you might say some of these more egregious bioethical abuses and proposals and manipulations of human life are outliers. I don't think we get away from that easily, but we need to highlight that those are not the beneficial biotechnologies that are really going to benefit not just individuals, but humanity writ large. But I think, in general, uh, biotechnology and the science behind it is focused on improving the human condition.
Mhm. And what ethical lines, if you had to summarize, do you believe society should never cross, even in the name of scientific progress?
I think that's very simple and fundamental. Anything that harms or destroys a human being at whatever stage or age of life would be unethical, and that we need to value each and every human being, each and every human life.
Hmm. And are there areas where our technological power is advancing faster than our moral wisdom or even our ethical wisdom?
I think we've come across a number of those particular things where somebody just decides to do an experiment and they don't think, again, that question, not can I do it, but should I do it? And that's where we then need those guardrails. We need, obviously, moral and ethical guardrails that people think about this. Everyone should be thinking about this. And that's actually where I think the public comes in very strongly. That, a lot of scientists may go off on a tangent once in a while, but they also are interested in
Mhm.
what society thinks. And of course, they're also interested in getting funding from the taxpayers in a lot of cases. So if the public is solidly formed in terms of what are correct bioethics and make their will known to the scientists and to the policymakers, I think those are some of the best guardrails we can have.
Mhm. And how should Christians and people of faith think about scientific innovation without falling either into fear or blind optimism?
And I think that's, again, a matter of education, of realizing what are the ethical versus unethical practices or potentials, and coming back to that focus on every human life, every human being. Being made in the image of God means that right there, they are valuable. And they need to not just have that, keep that inside themselves, but to talk to others about that basic principle.
Mhm. And with all of these different biomedical technologies we discussed, is there a danger that human bodies, organs, and tissues are increasingly being viewed as commodities or raw material for research? And how should we reverse course?
And, I think in any of these instances, these are largely just tools, and it's a matter of how they're used. And so it would be easy, whether we're talking about fetal tissue or stem cells or embryo research, to start talking about using these individuals and exploiting them. So it comes back to, again, our attitude about any human being, no harm, no destruction of a human life. And that moves us away from these dangerous pathways where we commodify and exploit other human beings.
Mhm. And you already mentioned that we should have guardrails in place. So what role does bioethics play before these technologies become mainstream, or hopefully that they don't become mainstream, that some of them never become mainstream?
Well, I think bioethics, first and foremost, does what you've been doing, is trying to get these issues out in front of people so they, number one, learn about them, become educated about it. And that is, of course, one of our main purposes for SALT as well, to educate people about it, get them thinking about these before these become an existential issue, and encouraging people not just to learn about it, to talk about it, to promote the ethical issues. So bioethics sets that sort of framework, that attitude, if you will, about where we should be going as a human race versus things we should stay away from.
hmm I mean, my question is, what kind of ethical training is being done in med schools as well as
Yeah.
in scientific programs? Because you would want our scientists and our doctors to have the framework in place before they ever step room
Yeah.
in their practice or in their lab, right? So can you give us a sense of what that looks like now, what the landscape looks like now, and what it should look like?
It's not as encouraging right now as it should be because in most instances in medical school programs or in science graduate programs, there aren't courses on ethics, if you will, or
Mhm.
even discussions. Or at most, it's, yeah, here's ethics. I shouldn't fake data when I'm writing up my paper, or I shouldn't cut corners on my experiments. And oh, yeah, I've got to write up that and for the human resource people about how we're respecting human life or getting informed consent. But I think it goes deeper and even earlier that even in elementary school and secondary school, we should be talking about these issues. And at the youngest ages, they ought to learn about the wonder and the value of human life. There's a push to have school kids just actually see what goes on during human development right now, whether that's a video, whether that's discussions about early human development. If we think about it, this is the most common experience that every human being has is going through human development. And
Mm,
so just our awareness of that and appreciation that that's a human being too, that education starts early so that by
mm,
the time they get to med school or graduate school, they already have formed a good attitude. And then they can add that, well, when I look at this experiment, now that's something we shouldn't do because that would violate human dignity.
mhm. And what happens when reproduction increasingly becomes driven by technological capability rather than the acceptance of human limits? I think that there's an issue with accepting human limits that's underlying this as well.
Yeah, and I think you've put your finger on a key problem there. It's back to, again, our attitude and our desires, if you will, that we all want the best for our children. But if that then becomes a matter of manipulating and even selecting, and if you select, there are going to be some that are chosen and some that are discarded, destroyed, and so on, that we have to realize that there are these kinds of limits and be accepting of humanity writ large.
Mhm. And for ordinary people listening in on this conversation, why should they care about what's happening right now in biomedical research?
Well, I think two things. One is, yeah, in terms of medical advances and things that you might be able to actually avail yourself of now or down the road, you want something that would be acceptable ethically, that doesn't rely on destruction of a human life, that this person has to die so that I can benefit. Now, that just does not help that individual who might be getting that treatment, and it doesn't help humanity at large. And again, if we talk about humanity in general and all human beings, if we accept that some are lesser beings or simply commodities that can be exploited, that attitude will end up pervading throughout society. So we need to maintain the highest standards and the highest goals for respect for human life.
That's perfectly said because if one vulnerable set of humans are not respected, then We are all in danger, essentially. Ultimately, what do you believe is at stake in the future of being human?
I think it's humanity in terms of being human. Again, this attitude, if we accept other human beings and we value them, that's also going to benefit us and our family and our close friends. So we need to, again, not just be accepting, but be valuing and loving all human lives.
Yes. Well, thank you, Dr. Prentice, for sharing your expertise. We need it desperately, and especially in this area of biomedical research that is moving so fast. And your wisdom and expertise on this is a gift to the world. And I hope that many people listen to this episode and understand what's going on this front, and that even scientists are listening
Yeah,
and putting a pause and thinking, what should I be doing with my talents and skills,
right.
and how can I be serving humanity instead of harming it? So thank you so much, Dr. Prentice.
You're welcome. Thanks for having me on.
For my conversation with Dr. David Prentice, President and founding board member of the Science Alliance for Life and Technology, and an internationally recognized expert on stem cell research, biotechnology, and bioethics, with nearly 50 years of experience as a scientific researcher, professor, academic leader, and policy advisor, here are 5 key takeaways from our conversation on biomedical research. 1. Just because we can make embryos in a lab doesn't mean we should, nor does it make them any less human. Dr. Prentice explains that researchers can now use stem cells, including cells derived from skin or hair follicles, to create human embryos in laboratories. These embryos develop, organize, and even form beating hearts, just like naturally conceived embryos, raising profound ethical questions whether scientific capability alone justifies creating human life this way. 2. Modern biotechnology is moving from healing disease toward engineering human life. Technologies like germline gene editing, also known as heritable gene editing, 3-parent embryos, lab made embryos, and embryo selection are shifting medicine away from therapy and toward manufacturing, modifying, commodifying, and optimizing future children. Dr. Prentice warns that this changes not only science, but our understanding of human dignity itself. 3. There are ethical alternatives to destructive embryo and fetal tissue research. Contrary to decades of public messaging, Dr. Prentice argues that embryonic stem cells and aborted fetal tissue have produced little to no successful therapies, while ethically sourced adult stem cells and induced pluripotent stem cells have already helped millions of patients worldwide. Scientific progress does not require the destruction of human life. 4. Designer babies and heritable gene editing could permanently alter future generations. Gene editing in embryos differs from treating disease in born patients. Dr. Prentice warns that germline or heritable gene editing introduces permanent genetic changes that can be passed down indefinitely with unpredictable consequences for future generations and potentially for the human genome itself. 5. The deeper issue is not just scientific capability, but how society defines human worth. From prenatal screening and modern eugenics to embryo selection and better baby marketing made by IVF companies, our conversation repeatedly returned to one central concern. When society begins selecting, designing, or eliminating human beings based on desired traits, we risk making quality of life judgments that redefine which humans are considered valuable or acceptable. Because truth matters and human dignity isn't optional. This is Bioethics Babe. Thanks so much for listening to this episode of the Bioethics Babe podcast. If you've enjoyed this content, please subscribe to my YouTube channel and your favorite podcast platform, and share with your friends and family. Also check out bioethicsbabe.com for the latest episodes and resources.